Hello everyone and welcome to this free webinar on bioinformatics for infectious diseases. In today's webinar, we will speak about genomic surveillance of SARS-CoV-2, specifically on the importance of genomic surveillance for novel emerging variants and consequences of large-scale vaccination on its evolution. We will also showcase some of the latest developments on the T-BioInfo platform designed for large-scale bioinformatics data analysis and used in virology studies, and introduce the upcoming Omics Logic training programs for those interested in effective utilization of bioinformatics for infectious diseases, immunology, and microbiology. This webinar is also part of the Omics Logic specialization track on infectious diseases, where we provide training on sequence variation and alignment, evolutionary and phylogenetic analysis, functionality and protein structure, transcriptomic analysis of host response. In this program, we will learn about the way the SARS-CoV-2 pandemic has transformed our appreciation of genomic and bioinformatics. Understand how genomic data analysis tools can help identify specific viral strains, understand multiple sequence alignment, phylogenetic analysis, and the significance of mutations in the context of viral protein structure and function. We will further discuss the viral genomes of COVID-19, genes, proteins, and the virology of the disease. We will also discuss how the data and the various analysis tools can help in the detection of viral genomes, compare and distinguish between viral strains, identify the impact of mutations on the functionality of viral proteins, and discuss the virology from the standpoint of data. In this slide, you can see a few news headlines on COVID-19 and its impact that is being felt all over the world. This clearly shows that there is much work left to be done when it comes to research. So before I pass on to our program speakers today, let me introduce you to what Omics Logic programs are, especially for those of you who are joining us for the first time. At Pine Biotech, our mission is to simplify bioinformatics for students, clinicians, and biologists, and we do this through our training programs known as Omics Logic. The Omics Logic bioinformatics training programs are running in 5 different regions with over 10,000 users around the world. Now, let me introduce you to the main speakers of today's session. We have Dr. Harpreet Kaur, who is our certified Omics Logic trainer and expert bioinformatician. She will guide hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is our Omics Logic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is the CEO and co-founder at Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners who are applying multi-omics integration bioinformatics to a variety of biotechnology and clinical applications. With this, now I would like to invite Dr. Harpreet to talk more about today's subject. Dr. Harpreet, over to you.
Thank you, Beepsa. Let me share my screen. Hello, everyone. My name is Harpreet Kaur. I have a PhD in Bioinformatics. Currently, I am curriculum developer and research consultant with Pine Biotech. In my current role, I am assisting participants to learn about data analysis, machine learning, visualization in application to the large NGS data used in the biomedical research. To provide hands-on training and to make participants familiar with the approach to design and complete a research project. We are continuously designing different research projects using publicly available data based on the studies published in high-impact journals. Thus, we have a collection of research projects where you can learn how bioinformatics can be applied in the diverse fields using publicly available data to drive biological and clinical significance from that data. So let me give you a glimpse of one of the projects on current pandemic of SARS-CoV-2, that is origin of SARS-CoV-2. So whenever a pandemic outbreak happens, like the current pandemic of COVID-19, there are several questions that needs to be answered. So these questions can be of following: where did the novel coronavirus come from? Which animal or what location? How did it start infecting humans? To understand and design the vaccine against any virus, we need to find answers to these questions. So towards this, in the year 2020, Anderson et al. Published a research letter in the journal Nature Medicine, in which authors studied the proximal origin of SARS-CoV-2 based on the NGS data. And they also find some of important mutation in the spike proteins that are responsible for its enhanced pathogenicity. So based on this study, we designed a project. In this project, we explored this pathogen coronavirus itself, as well as the disease caused by it, that is COVID-19. So here you will learn how you can find its origin, how much it is related with already existing human coronavirus strains based on the multiple sequence alignment and phylogenetic analysis, based on the data that is taken from publicly available repositories. Next, you will learn about what are important components of the virus through which they will enter into the human cells. We will also look into how much it is different from other existing coronavirus strains in the human. At last, you will see how you can identify important signatures that can significantly different under a different therapeutic treatment or untreated conditions. So as we know that currently there are various public repositories that maintain NGS data for the SARS-CoV-2. For example, one of the repository that is NCBI virus. So NCBI continuously collecting all the viral genome from the pandemic and associated viral sequences of the SARS-like viruses in this repository, which is also known as NCBI virus. So during this upcoming workshop, you will learn how to obtain NGS data for SARS-CoV-2 from these public repositories. So after downloading the data from the NCBI, next you will learn to perform phylogenetic analysis to understand its genetic origin. And you will also learn how you can compare it with other existing strains from the human samples itself based on multiple sequence alignment and phylogenetic analysis. Subsequently, you will learn how you can perform multiple sequence alignment to understand the mutational changes that are taking place in SARS-CoV-2 in comparison to the other strains from different animals and human samples. Further, you will also see how you can analyze multiple sequence alignment to understand the differences and the mutation among different samples at nucleotide levels and amino acids levels. You will also learn what are the key mutations in a specific region that result in the enhancement of the pathogenicity of this virus. Next, you will also learn what are important protein components in the virus and the host that are responsible for its pathogenicity. For instance, how ACE2 receptor binds to the spike protein. So here you will also learn how to visualize structure of this particular spike protein and its complex with ACE2 receptor. In the next part, you will also learn how you can obtain transcriptomics data for the patient that are affected with the SARS-CoV-2 from the public repository like GEO, that is Gene Expression Omnibus. Here you will learn to explore this data to find out important transcriptomic signatures that are associated with the particular drug treatment, like your ruxolitinib. Similarly, you can analyze such data to understand how the virus causes infection and What are some of immune response differences between acute and mild COVID-19 disease condition? So now let's go over T-BioInfo platform to see and how to let me perform a demonstration of phylogenetic analysis to understand its unique reason and explore the results. So if you want to follow along, go with me to the T-BioInfo server and do the steps like I will do. If you do not have account, you can register on the learnomics.com and use the same credential to log in. So now let's go over T-BioInfo server. So let me first share here. You can register yourself on this site. So this is our T-BioInfo server, which we thank you, Vipsa, for sharing server link. So on this platform, you will find various sections to perform different types of analysis using NGS data, like transcriptomics section, genomics section, then there is a dedicated section on mass spectrometry and structural biology, then this is data integration and data association. Beside it is here we have also a dedicated section on virology. So right now I am going to here this section under the data association, you will find this section that is phylogenetic tree. So here you will find this drop down. So this drop down we have example project example pipelines. So here I am taking the third example that is demo SARS-CoV-2 reason. So if I click on this and I will direct it to this interface. So with the start, as this is already example project, so we have already uploaded our data from the SARS-CoV-2 and the three uh, data from bat coronavirus and pangolin coronavirus and bat SARS. So then after uploading our data, the next step is to perform multiple sequence alignment. So here all the sequence that have taken from different samples get aligned. So after the multiple sequence alignment, then using the translation codons, so then we are converting them into the amino acid codons. So then you are selecting positions on the MSA. So there are the different positions. So this module select only specific codon position for further phylogenetic analysis. So then which phylogenetic tree method that you are using, right? Like here, there are different methods like APE, Beast Coalescent constant size based method, Beast speciation Yule process method, Beast speciation birth and death process method. So here we are using all the methods. So here we choosing where we can generate phylogenetic tree using all the methods. So then here then we are using the tree space method. So what it does that it will generate eventually median tree based on all the methods. So then we are with and clicking on the end, we will able to complete this pipeline. So once we able to complete the pipeline, under here the output files. So if you click on this download pipeline outputs, so you will get various types of outputs. So here you will find phylogenetic tree based on the PDF files. So similarly, we have also phylogenetic tree, median tree that is generated by tree space method. So if you download these files, so now let me show you what it contains. So you will get the phylogenetic tree like this. And beside, you will also get an output like here, that is full table TSV, which contain information related to the mutation. Apart from that, you will also have a sequence that is a line sequence in the cluster using the cluster method. So you will find out different outputs. So here, let me just show you this output. Specifically, we have also further analyzed these outputs. So here, what you will observe. So you will find the multiple sequence alignment. Then based on the multiple sequence alignment, we will get this phylogenetic tree. So this phylogenetic tree clearly shows that human SARS-CoV-2 genome sequence is more close to the pangolin coronavirus in comparison to the bat coronavirus and bat SARS. So if you are interested in getting started with such analysis on your own, I recommend you to going to this project on Learn Omics and practicing these exercises before starting to analyze other data sets related to the SARS-CoV-2. So let me paste its link as well. So on this platform, this project is divided into the different lessons depending upon the objective of each study. So now I would like to pass this session to Ilya Brodsky to speak about analysis of variant taken from clinical and environmental samples, where we will find variants of the concern and link them to clades of reporting. So over to you, Ilya. Meanwhile, if you have any questions, we will address your questions in the chat box itself.
Thank you, Harpreet. And I will kind of pick up on where she left off. So my name is Ilya Brodsky, and I will be speaking about what happened with the SARS-CoV-2 genome since the pandemic continued for now over a year and a half, and what are some of the major concerns now as it is continuing to evolve under a unique set of evolutionary pressures that are specific to different geographic regions and different populations. And so we'll see how genomic data analysis of SARS-CoV-2 genomes in humans that are continuing to be deposited on NCBI and GIS-AID can help track emerging variants of concern, and what are some of the ways that you can analyze this data, and what are some of the insights that you are expected to find. So we'll talk briefly about linking this original evolution with specific variants, and then thinking about these variants of concern that are in fact collections of variants. They're not just single mutations, but collections of variants that we should be paying attention to. So a lot of what I will be speaking about is an introduction to the upcoming training program that we will do, and this will be a program that will last 5 days. So we will get a lot of the in-depth training in the program, but today it's just more of an introduction. So in the program itself, we will learn to identify specific viral strains, understand multiple sequence alignment options, see phylogenetic analysis and how it can be used to detect different clades, speak about the significance of mutations in the context of viral protein structure and function. And then we will also discuss the viral genomes of COVID-19, the specific genes, proteins, and the virology of the disease. So then we will talk about how can you practically use publicly available data, open source tools like R, to generate a report and prepare yourself in case you have to analyze data or potentially ask questions about the nature of publicly deposited data sets in the context of the emerging variants of concern. So before we get started with the current variants of concern that we will speak about in just a moment here, let's look at one particular mutation. So this particular mutation appeared early on in the pandemic, and it is the G614 genotype. So it was first reported in February, and you can see the emergence of this mutation in 33 sequences deposited on NCBI. So it was really observed at low frequency. In March, it was about 26% of the total available sequences had that mutation. But then it continued to increase rapidly. And so by April of 2020, it was 65% of the sequences had that mutation. And in May, 70% of those sequences had it. So it really indicated that this mutation had a transmission advantage over viruses with D614, right? Because the mutation was from D in the position 614 in the spike protein to G. Korber et al. Noted that this change also correlated with increased viral loads in COVID-19 patients. But because this change is also associated with the mutations in viral NSP3 and RDRP proteins, the role of the spike protein in these observations remained undefined. And so some studies took a look at the D614 gene mutation and how it alters the properties of the spike protein in a way that could impact transmission or replication. So researchers wanted to assess the role of viral entry and the mutations' increased ability for cell entry. So experiments with both viral strains, so strains G614 and D614, showed that infected HACE2-293 T cells showed approximately 9-fold higher efficiency in viral entry for G614 compared to D614. So an interesting question arises on why viruses that are carrying the more stable G614 mutation appear to be more transmissible without resulting in a major observable difference in disease severity. So it was possible that higher levels of functional spike protein observed with G614 increases the ability of host-to-host transmission, but that other factors limit the rate and efficiency of intra-host replication. Alternatively, the loss of variant-associated spike protein observed with D614 may be compensated by greater fusion efficiency with the destabilized S protein when the next target cell is adjacent to a host tissue. So what you're seeing here is that this particular mutation, which you can see alters kind of this region, specifically where the two domains of the spike protein are connected, could be raised with the kind of continuation from where Harpreet started, which was to speak about the adaptation process. So as you go through that project that describes the adaptation between bats and humans, you will discover that a particular segment of the spike protein is clearly changed between bats, pangolins, and humans. And that is this furin cleavage site, which allows the S1, S2 boundary to be cleaved in virus-producing cells. And so this particular polybasic cleavage site is cleaved by TMPRESS2 and allows greater stability and entry into the cell. So this D614 gene mutation may have emerged to compensate for this acquired furin site. And so the special focus on the spike protein led to further studies and models that could establish a clear link between phenotype, genomic variants, and to study the biology behind this link. But that's why even before we start this training that will begin next week, it's important to kind of go through some of the basics if you're still not familiar and understand what are how is the genome of the SARS-CoV-2 organized? What are the different proteins and how are they encoded in these genes? And then what is the structure and function of the particular domains of the spike protein? Now, the variant containing the D614 gene mutation has become predominant genotype of the wild type SARS-CoV-2 worldwide since approximately the middle of 2020. The mutation likely increased the affinity of the virus to bind to the human receptor ACE2, resulting in higher infectivity and transmission rates, but potentially not increasing the mortality or the severity of disease. Variants can also become extinct due to natural selection. For instance, in June 2020, a concerning SARS-CoV-2 variant thought to present a decreased sensitivity to the immune response was detected in mink in the Netherlands and Denmark. However, this variant was declared extinct just a few months later and is actually no longer in circulation. So since then, the collection of genomic data continued, and a lot of the analysis was focused on determining distinct clades in viral phylogeny, tracking how it changed over time. Now, phylogeny is different from individual mutations because we're speaking about a mutational signature that helps us identify a distinct population that you can see here colored in different colors. And you can see that these changes kind of appeared over time, separating into more and more distinct regional selection that is associated with clades that have become extinct or diminished in prevalence, and the ones that have become more established and more consistent in the greater population. Bioinformatics methods that find individual mutations like multiple sequence alignment can be combined with evolutionary models that track changes in the genomic sequence over the whole genome and then link them to a particular evolutionary process. So these models, and Dr. Kaur showed some of these models earlier, they fit to thousands of genomes, and they can reveal this dynamic tree of the pandemic, which shows that the virus is continuing to change pretty rapidly with novel distinct clades that are separating from a common origin over the course of the pandemic. So by the end of 2020, there were approximately 12 distinct clades, and their number is continuing to grow. As you can see, there have been 3 new clades defined just since November 2020. And the clade proportions are likely to be unreliable due to the bias in sampling between various countries and even outbreak regions where sampling is done in much greater numbers, right? So for example, we know some countries are just doing a whole lot more sequencing. So the clades, and you can see down here, some of those clades and how many have been reported during different time periods. So here from January to February, 3 clades, 5 clades between February and May, and then 7 to 8 clades between May and the end of 2020. And so here we now have a bunch of different clades that you can observe here at the very end. So by the way, this is NextStrain, so you can navigate to that website to find all of this information there. So instead of individual mutations, clades are essentially based on multiple mutations occurring at the same time. They can be found in the spike protein, but also in other genes. So this makes it a bit more hard to investigate the biology behind these changes, as the complex molecular mechanisms of multiple mutations can have an effect on are hard to replicate in controlled experiments. But as a result, we came to this notion of variants of concern. So these were variants or combinations of variants that appeared in a non-random way. And this highlights the significance of genome tracking as we continue with this pandemic. So here you can see some of the prevalent mutations, and you can see how they are distributed, for example, in the US. And here are some of those mutations associated with the 20I variant. So the 20I variant is the UK variant across the spike genome. So overall, these clades are represented by mutations that affect different proteins and their functional domains. But you can see here, this is if we take a collection of sequencing data, collected genomes from various locations, and we just analyze them, we can map the mutations on the spike protein, and you can start seeing how the specific regions of this protein are non-random. You can detect these changes appear to be more likely to happen in specific regions. And so now that we have access to all of this data, anyone can actually access this data and start asking the question, how can we detect these mutations? How can we associate these mutations with phenotypic data, for example, about time or about geographic location? And what do we make out of these individual mutations? So currently, there are 3 primary variants of concern, which are named based on the country where they were first identified. So there's the UK variant, B117, South African variant, B1351, and Brazilian, which is P1. So these variants have mutations which are thought to make them more transmissible. There are also early data to suggest that UK variant is associated with a higher mortality risk than the wild type SARS-CoV-2. And the mutations in these variants lead to small changes in the structure of the spike protein, but also in other proteins. But this protein is used by the virus to enter human cells. Now, the spike protein is also a part of the structure of the virus where neutralizing antibodies bind to block the SARS-CoV-2 infection. Consequently, these mutations can affect SARS-CoV-2 in an advantageous or disadvantageous way for the host or for the virus. The most concerning is how these mutations will affect efficacy of vaccines or the protection, the long-term protections that they offer, but they're also a reflection of how viruses evolve under very different immune pressures, because treatment and vaccination has already started, and the proportion of affected people with SARS-CoV-2 is also changing. So that means that the environment that the virus is replicating in is changing. So here you can see some of the hypotheses about how this virus is going to affect effectiveness of vaccines and long-term protection that they offer. So you can see that basically the vaccines that we have today, including the Pfizer vaccine, Moderna vaccine, AstraZeneca, and others, are all based in part on the spike protein. And the spike protein that contains these mutations then trains our immune system, and the immune system can recognize this protein based on binding of specific antibodies. And so some of these mutations occurring at the binding sites are likely to have a profound effect on how vaccines are going to be effective and whether new vaccines are going to be needed as a boost shot for those that are going to continue and face these new infections. In fact, today we are probably all concerned with this tremendous spike in infections in India. So here you can see a report. So this was already an alarming report from March. You can see that everybody was looking at this trend, but you can see at this report in April how the report in March, right, March 22nd is about here, how since then the acceleration of infections has been practically exponential. So how does this reflect these variants of concern? So over 10,000 samples from 18 Indian states showed that up to 771 cases of known variants, 736 of the UK, 34 of the South African, and 1 Brazilian have these mutations. So some people have started to hypothesize that there's a double mutant. And double mutant, what they mean by that is that there's a separate lineage that is developing in India with amino acid mutations L452R and E484Q. These are positions of amino acids in the spike protein that are occurring together. But scientifically, the term double mutant actually makes no sense. SARS-CoV-2 mutates all the time. So there are many double variants all over the place. And the variant in India is probably a representation of many different variants that are occurring simultaneously or separately in different substrains or subpopulations of the virus. So for example, other variants of concern like B1617 from the UK doesn't contain just 2 mutations, but it's more than a dozen. So for example, there is a mutation L452R that's also present in the dominant strain in California. B1617 has a similar mutation called E484Q as the one present in the variants first detected in South Africa and Brazil. And so that mutation is called E484K. So it's the same position, but different amino acid substitution. So there are 2 famous mutations associated with this Indian variant, but there are also about 11 other ones as well. And so the question is then, is this type of a strain more concerning? Is it more contagious? Is it going to be more deadly? Preliminary evidence suggests that this new Indian strain is more contagious than previous strains of the virus. A study published recently found that the L452R mutation may enhance the ability of viruses to infect human cells in the lab. The variant in California, which carries the same mutation, is about 20% more transmissible than older strains of the virus. And so now, as this strain continues to spread, in the past few months, it is seemingly becoming the dominant strain in the state of Maharashtra. Sorry if I mispronounced that. But the bottom line is that these strains are rapidly emerging. They're very different. They can have different phenotypes. And the key is to continuously collect genomic data to be able to identify what we can find and then ask the question about what might be the link to a phenotype. Now, importantly, these mutations is what's been reported, but recently there have been some additional reports that come from the efforts of researchers around the world as they reanalyze data that is found in the public domain. So here's a preprint that appeared on bioRxiv recently about large deletions that consistently are found in the spike protein and might have a profound effect on vaccination as well. So this figure right here is the identification and characterization of recurrent deletion regions in the SARS-CoV-2 spike protein. So here you can see that this was found based on the GIAS-8 sequences. There are 4 clusters here with recurrent deletion regions. And these deletions are of various lengths. So you can see the position in the genome and the length of these recurrent deletions. So these types of novel insights are not being reported at all, but they could have a profound effect on what is actually happening below the consensus level. Okay, so now that we got to this point, let me kind of briefly introduce the significance of this genomic understanding of viral trends as the pandemic continues to evolve. In fact, there is now a national open genomics consortium for COVID-19 response. And so the Advanced Molecular Detection Program is established to coordinate SARS-CoV-2 sequencing. And so this is an effort called SPHERES, and the SPHERES collaboration includes scientists from clinical and public health labs, academic institutions, and private sector. SPHERES it was established to strengthen COVID-19 mitigation strategies, and one of their efforts is large-scale rapid genomic sequencing that will allow public health experts to monitor important changes in the virus as it continues to circulate, gain important insights to support contact tracing efforts, provide crucial information to aid in identifying diagnostic and therapeutic targets, advance public health research in the areas about transmission dynamics, host response, and evolution of the virus. And this consortium, which is led by the CDC Advanced Molecular Detection Program, over the past 6 years has invested in federal, state, and local public health labs to expand the use of pathogen genomics and other advanced lab techniques to strengthen infectious disease surveillance and outbreak response. The aim of this consortium is to actually accelerate the use of genomic data and identify these trends, translating them to public health insight. And so that's why we thought that it's important for us to discuss briefly what are some of the opportunities to identify mutations and variants and link them to these efforts. So first of all, I'll briefly speak about consensus level analysis of variants. So this is analysis of how we can find FASTA genomes on NCBI and GIAS-8, and performing multiple sequence alignment, find mutations that are most prevalent in a sample. Now, there, one of the important opportunities is identification of novel prevalent variants. So we can find variants that are already established in the population and ask the question, what is the consequence of that variant? Then there's an opportunity for sub-consensus and low frequency variants. So what I mean by that is when we take FASTQ files from raw next generation sequencing, and we're asking the question, what are the minor frequency variants? What are the below 50%, below 20%? Maybe here we have an opportunity to identify some of the emerging variants that might become established in the near future. And so asking those questions and having the tools to ask those questions is an important part of what we're going to be talking about in this full training program. Now, sometimes these variants of concern, and I'll speak about this in just a second, are not just detected in clinical samples. In other words, you have a lot of patients. These patients are in hospitals. They're already with clinical symptoms, and there you have a lot of genomic data. But before people get to the hospital, before they have clinical manifestation of disease, where can we find information about what kind of virus do they carry? Because they probably are the major spreaders. They're not treated, they're not cared for, they might be even asymptomatic. So they might have a lot of contact. And so the question is, what can we find there? And some alternative techniques could be done to detect those, and those would typically be in low quality samples. And I'll speak about that in just a second. And then we're also talking about tracking spread and emergence of variants in densely populated areas. So the question, how can we kind of preemptively track those variants, and where can we find data for that? So to speak about that, I wanted to talk about this new opportunity. So as you can see here, the National Institute of Health grant launches study to track SARS-CoV-2 in wastewater. And you can see this opportunity is posted on the RFA, which is to essentially collect wastewater samples and track variants in those samples that are typically degraded RNA at low coverage, because we are recovering only portions of the RNA, and then still the ability to be able to find and report on particular variants of concern. So here, recently this has become public, so I'm happy to share this. So the Division of Biomed at the Louisiana State University School of Veterinary Medicine has been analyzing wastewater samples from different locations in East Baton Rouge, as well as LSU dorms, in collaboration with Department of Civil and Environmental Engineering for detection of viral genomes. This strategy has already helped the university address transmission by getting a head notice about a potential infection spreading on campus. qPCR analysis is currently being used to detect viral genome units in wastewater collected from near dorm sites, as well as wastewater processing stations around East Baton Rouge. Recently, novel variants were identified in the SARS-CoV-2 virus that increases transmission and potentially can help the virus evade the immune response. These have been reported in the UK, South Africa, and are now expected to make their way into the US. Getting an advance notice on such variants' appearance in Louisiana, and especially among students living on campus, is a major part of being prepared with a mitigation strategy. So the LSU Biomed conducted a next-generation sequencing study using Illumina MiSeq platform. Biomed has partnered with Pine Biotech to analyze the sequencing data. And so what kinds of challenges are we talking about when we are analyzing this kind of data? Now, I'm not going to be showing you any of the data itself because it is private, but there are other publicly available data sets out there from similar studies that can help us address the question of quality of data, reporting on specific variants, and identification of how impactful this could be by linking it to known variants, or by asking the question, what are the new variants and why are they important? So one of the first things that we talked about is this consensus level reporting, where we can identify only the positions that have some consistent variants that could be of importance for this kind of preemptive reporting for the public health information and mitigation strategies. And here we can see how many, for example, mutations are present in a sample, or how many mutations are present in a particular gene, as well as the positions that are present there, asking the question, well, what are the positions that are known to be associated with these variants of concern? An important part here is developing a pipeline that could be automated and could provide important tools like pre-processing with Trimmomatic, for example, and identification of all of the present per nucleotide mutations, which, as you can see here, a brief comparison, and we'll do this comparison together in the upcoming training program, has an impact specifically on some of these low coverage samples, as compared with no pre-processing, PCR clean only, or Trimmomatic and PCR clean. So you can see some of the differences here in the low coverage, which means more degradation, right, essentially. And then the second part of it is, how can we report on the actual variants that we have found? And this is an example. So this is a report, a demo report provided by Cosmos ID, which is a company that is commercially offering these kinds of reporting based on clinical samples. So you can see that essentially what we will learn to do in this upcoming training, which I think is a very important part of dealing with this kind of genomic data, is actually preparing a visual report that you can share with biologists and clinicians, as well as public health officials, kind of incorporating the important information that there is in the data itself. And so here you can see a critical component, obviously, is coverage, because with higher coverage, we have more reliance on the data that we have. With low frequency positions, it's especially important because they're only a small proportion of the reads that we will find. And then actual reporting for the variants. So here, each one of these variants of concern is a combination of anywhere between 5 and 12 different mutations. Those mutations have different proportions. So here you can see the proportion of these mutations that are found in different samples for a particular variant. And so Preparing these reports means having access to the information about variants, analyzing the proportions, and then being able to call on a particular variant with high enough confidence. So we'll talk about data preparation techniques. We'll talk about how this data gets processed, and then we'll talk about how to evaluate some of the variants that you are finding to be able to make that information accessible to others that might not go into the depths of the bioinformatic analysis that you have performed. So as a part of this training, what we want to do is we want to equip others that might be dealing with this kind of public domain data, or might be dealing with collection of new samples, maybe from wastewater or from clinical samples, to be able to effectively and efficiently analyze it and provide reporting that is meaningful and reliable. And so here we will talk about different methods, different pipelines, different kind of annotation resources that you can leverage to be able to conduct such analysis. Now, this is also important for those of you that might not be involved in the actual collection of samples and analysis of samples. And the reason is because you can also acquire important skills. And so the skills that you will gain from here is to read literature and understand methods of preparation of this omics data, databases that have the data available that you can take and you can analyze, methods of pre-processing and processing of this data on large big data processing cloud-based platforms like the T-BioInfo platform, and then some skills in R, which we will use to actually prepare the report from a table that combines nucleotide coverage and amino acid information that allows us to ask questions about the data that we are analyzing. So now I would like to pass it on to Dr. Mohit Mazumdar to speak a little bit more about the program that is coming up and some of the other training opportunities that we're going to be offering in the coming weeks. So Dr. Mazumdar, over to you.
Thank you, Ilya, and hello, everyone. So next, we will just show you that what all we will be covering in each of these sessions. So for all who are joining for the first time, my name is Dr. Mohit Mazumdar, and I will be one of the co-mentors of this program. So the first week of the program, we will be starting the training on May 3rd, and it will go on for 5 sessions. So these are the 5 sessions that we have planned. So it would include starting from, we will go over each of these topics you heard today and in great details with hands-on practice. So specifically, we will discuss the trends behind the emergence of SARS-CoV-2 variants, and then we will talk about the genomic surveillance, where we will be discussing data collection, sequencing, and bioinformatics analysis. And then the third session will be downstream analysis of genomic DNA. So we'll be learning about the evolutionary studies, phylogenetic analysis, tracing the origin, and then reporting. So that reporting will be also a part of the fourth session, where the Ilya discussed like the custom R scripts, and then about the vaccination. To tell you in detail about each of this session, that how this will be spanned and how we will be carrying this session throughout that time. So the first session will be on Monday. So in the first session, we will be talking about the nature of emerging variants, why they are causing concern, and what might be the driving force behind the observed clades or substrings of this virus. So as this virus continues to spread around the world, we will review some of the latest literature and find dataset examples from these studies. So that will be for the first session, which will be on May 3rd, Monday, at the same time. In the second session, we will talk about how data is collected. That will include overview of next generation sequencing, RT-PCR, and other surveillance methods for detection of viral particles. So sequencing of their genomes and detection of variants will also be there. So we'll also speak about established protocols like the Illumina Arctic protocol that is used for the full genome sequencing, and PCR primers for variant tracking. So this will help us organize data by type of sequencing and then prepare for the analysis. So that will be what we will be covering in the second day on Tuesday, May 4th at the same time. For the third session, we will discuss methods for bioinformatics analysis, like data processing, evolutionary analysis based on multiple sequence alignment, then phylogenetic analysis, phylogenetic tree reconstruction, as well as concepts behind the variant calling methods. So that will be on Wednesday, May 5th. Then on the fourth session, which is on Thursday, we will have a hands-on session to load files from NGS pipeline into R, and then use custom scripting to produce a report on variants identified as consensus or sub-consensus level of detection for major and minor variants. So in this session, we will touch upon important considerations for variant calling, and then we will also be working on reporting various clinical grade and degraded quality of RNA sequencing that Ilya just discussed in today's session. So for the last session, which will be on Friday, so this will go on for a week, we will discuss some of the future expectations, as this type of data is likely to grow in abundance as we all are generating data. So when many countries around the world are investigating billions of dollars in sequencing variant detection, so what could be the vaccination and other interventions for SARS-CoV-2? So that will be the last session on Friday. So in this entire program, you will be accessing some of the resources which will involved in the program. So to talk about the resources, let me now pass it back to Ms. Beepsha, who will guide us through the program registration as well as the access to various resources that we just discussed today. So over to you, Beepsha, if you can go through the program resources, and then maybe I can show them the upcoming programs that we have planned.
Sure, Dr. Mohit. So I will also go over the upcoming programs a bit, but let us talk a little more about the program resources and address some of the questions that everybody has here on the chat. So Ilya is addressing those questions that you have asked. So he would be addressing those. Now, again, for everybody who is joining us for the first time, and also for everybody who has joined us after, say, a while, the new platform, which is a Learn Omics Logic platform, is now linked to the T-BioInfo platform, which means that if you create an account on learn.omics logic, then you automatically get a demo account on the T-BioInfo server platform. So you can use the same login IDs for both the platforms. So if you are there already on the platform, please sign up here. And I'm going to be taking everybody through a number of free resources that you can take a look at and use. So I have already created an account here. You can create your account using this. I will just sign in. And once I sign in, I am brought to this page where I can find different projects and different courses on multi-omics topics, on R and Python for bioinformatics. So please feel free to take a look at all of these projects. And today's session, Dr. Harpreet went over this project with you. So this is the link of the project here. And I'll show you also here how you can access this. So this project is the second one, COVID-19 origin and pathogenesis of SARS-CoV-2. So if you have some of those questions, you can go ahead and start by studying this project. That will give you an overview of what is being covered and also help answer some of the questions that you may have. And next, we have another course, which I highly recommend everybody who is joining into bioinformatics for the first time or wants to get started with bioinformatics take. This is the Introduction to Bioinformatics course. And there's a video that you can watch, and there are also different lessons on how bioinformatics is applicable in areas of, say, agriculture, healthcare, also in different other areas of research. So please go ahead and feel free to read through this and take the courses. And besides these courses, you can actually go through any of the courses that are listed here. As you're going through the courses here, you would notice that there are some courses which are marked with a star. All the courses that are marked with a star means that they require a subscription. For all of our participants who are joining us in any of our upcoming programs, you will have full access to the platforms. So please make sure that you're utilizing the same email ID to log in with which you register for the program, so that you are given those access. And for anybody else who are only interested in staying at the free level of the program, there are a number of courses which are currently available for free for you to go ahead and review them. So these are the resources that we'll be using. And of course, we will be using the T-BioInfo platform here. And if you are having any problem, again, in accessing the T-BioInfo platform, all you have to do is create an account here and log into there. So with this, I would want to ask Dr. Mohit to share with you again about our upcoming programs. And then I have a poll for everybody where I would want to learn more about your interests as well. So Dr. Mohit, over to you.
Thank you, Bibsha. So the Omics Logic summer programs that we have planned are skill-driven research-based training programs. So so the summer of 2021 is surely different from what we have witnessed so far. We are into the second year of the pandemic, and all the human lives have been affected one way or other. So with many of us working from home, we wanted to make sure that we do our best to bring students and researchers opportunities to learn from great educators, to share vision about the future of data-driven research, and best of our Omics Logic project-based training programs. So one of such training program, which I'm really excited about, is biomedical drug data science and drug discovery. So this is a 4-month training program allowing students to participate and work on genomics, transcriptomics, data, and learn data science tools and algorithms to apply to both sequence and 3D structures. And this training is an opportunity for everyone or anyone to learn about to learn from a panel of experts and mentors who have years of experience working with pharmaceutical companies and startups. So they will provide important insights about the role of data, omics analysis, and industry-specific challenges that are important for a career in this field. So the mentors will teach along with our experienced Omics Logic team to guide you help find answers to your questions and gain hands-on experience. So in this program, this comprehensive and intensive research training is divided into 5 phases. So students and researchers interested in working on their own projects to learn about different omics and apply bioinformatics tool to the chemical space, and to gain hands-on experience on modern data drug discovery pipeline. So you can learn more about it, and Viksha, if you can share the link for the pre-registration and the registration. So this is one program. So another program that we are looking forward to is Bioinformatics for Infectious Diseases. So what we discussed today is one of the specialization tracks, and the major specialization is about learning about these infectious diseases. So Bioinformatics for Infectious Diseases is an online training program which is designed for biologists, clinicians, students that are interested in virology, microbiology, and immunology, and would like to learn about the use of bioinformatics and big data for infectious disease research, diagnostics, as well as drug and vaccine development. So this program will have several examples of disease projects with curated data sets, as well as guided analysis sessions to see how bioinformatics can be used for infectious disease research. Another program that we have is Genomics in the Virtual Lab. So Genomics in the Virtual Lab is a special opportunity to learn about genomics and big data analysis from a world-renowned scientist and a team of bioinformaticians working on cutting-edge big data challenges. And we will be learning by Dr. Sona Vasudevan. So she is a professor of biochemistry and molecular biology at GUMC. And she will present the disease-associated projects that program participants will be able to learn from and see how genomics is used at the intersection of data science and biomedicine. So this will start in June. And another one is Bioinformatics for Precision Medicine. So we will have all of these programs in May 2021, June 2021, and July 2021. So they are a part of the summer training programs. So Bioinformatics for Precision Medicine is 1 month program where where you can learn about the role of data in basic and translational biomedical research. So here we will be using both chronic and acute infectious diseases, and as well as oncology research. And we will see that the increasing significance, like how we can apply NGS technology, and that could help, and the omics data to come up with diagnostics and also treatment selection. So last one is the research fellowship program. So the research fellowship program is a program that is built for someone who have already either gone through the Learn platform and has some understanding about the T-BioInfo platform, or someone who has done one of these programs. So research fellowship program is designed to provide support and guidance on development of an independent research project. So for an independent research projects, we have a time frame or schedule for 3 or 6 months. So either you can do a 3-month research fellowship, or you can do a 6-month research fellowship. So during this time, the participants meet with us on a weekly basis to share their progress and get feedback from the program coordinators. And then they can also request one-on-one session with mentor on a weekly basis, or meet in a smaller focus group. So here we work on group projects where a number of interns are working on a specific problem that could be related to neuroscience or infectious diseases, or cancer biology, or cancer precision oncology. So as well as agriculture. Uh, so as we learn from all of this and we come up with different projects, the idea is to be able to show that outcome as a poster or as a publication. So you can also learn about all of this upcoming programs and opportunities if you join us on our social media groups, provided that you use LinkedIn, Facebook, Instagram, or Twitter. So this is our LinkedIn group, where we keep on posting about job opportunities, as well as about the ongoing program and literature that is coming up, which could help you associate, NGS data to bioinformatics analysis to different kind of research areas. So here you will find details about the blog posts that we are writing about different programs, as well as analysis strategies for example, malaria research, COVID-19 research. Similarly, we have a Facebook group where you can also join us, where we keep on posting about the new members, giving them insights about the upcoming sessions, which is happening around the globe in this online world. So where you will also find the researchers who completed their analysis and been able to either publish a video presentation, or a poster presentation, or a preprint, or going ahead for a publication. So you'll find all those information if you join our groups here to get regular information all the time. And with this, I would like to pass it back to Deepsha to maybe answer any of the questions related to registration, and how could you register for the program, for this short program, and how could you go about for the other opportunities that we just showcased. Thank you.
Thank you so much, Dr. Mohit. So I will go ahead and share my screen again. So if you are already registered on this site, then you would have received an email with the payment link that you can use. In case you are not able to pay via PayPal, please reach out to me so that I can offer you any other options that we have for payment. And all you have to do is fill out the form. It will directly take you to the payment page here. If you want to pay again, you can fill out the form again and it will take you to the payment page. And if you are not able to pay via PayPal, then you can again reach out to me. So my email ID would be marketing@pine.bio. And the cost for this 1 week workshop is $50, or in INR, it is 3730 INR approximately. So if you have any questions regarding this, again, please feel free to reach out. I've posted several links in the chat, so I would want to show you what these different links mean. And the first link, as Dr. Mohit was speaking, is about all of our upcoming programs. So if you are interested in any of our upcoming programs, and we keep on updating this page with all of the information. So we have the Research Fellowship Program, which is a program that we run all throughout the year. It's available for either 3 months or 6 months, and you can join this program as well. Then we have this upcoming program that we spoke about today. We will also be starting genomics in the virtual lab in June. So this would be guided by Dr. Sona Vasudevan, as Dr. Mazumdar has pointed out. And we are currently on the first phase of the Biomedical Data Science and Drug Discovery program, where we are going through genomics for biomedical drug discovery. So if you're still interested in joining this part of the program or any of the other parts, so the next program would be transcriptomics for biomedical drug discovery, then data science, and finally chemoinformatics. If any of these are topics of your interest, you can join either the full program or you can join one part of these programs. And all you have to do is go ahead, fill out all of your information, and you can choose from here whichever program you want to join in. So again, I will share the link in the chat box so that you can look here, and you can also bookmark this page so that anytime you want to join, you can go ahead and take a look at it. So Leela, the cost of the program is $150 USD. That is a beginner cost, but we would have discounts for the program as well if someone is joining right now. So please reach out to me for all of those questions. If you are interested in joining the Biomedical Data Science and Drug Discovery program, just write to me and I will be able to send you those details. And as Dr. Mohit showed you, we have these groups on our social media networks. And of course, we are going to be sharing here a lot of things which are informative for our community members. But if you note here, the members of our group, they are a diverse number of people who join this group, and they come from different fields of research. They are students, we have researchers, so you can actually go ahead and join this community and be part where you can continue to interact with everybody and build your own networks and connections. So I welcome you all to go ahead and join. And now I have a special poll that I hope you would go ahead and take. So I'm going to be just sharing the poll on the screen. And the poll has a few questions, so it would be wonderful to hear what your thoughts are. So the first question is if you are interested in researching on SARS-CoV-2. So if you are, please put down yes or no. Was this webinar helpful for you? The third question is, would you like to join our upcoming 1-week workshop starting on May 3rd, which is the one that we have discussed today? And if you have any other research interest other than infectious diseases, we do believe that you may have other interests. So if you have any interests which are not mentioned here, please post those in the chat box so that we are aware of the different interests that you have. And you can also reach out to us regarding those as well. These are some of the common topics of interest that we get from our community. So we have posted them here. And again, for any questions that you may have in terms of research, in terms of bioinformatics, please write to us at any time, or you can also post those questions in any of our social media groups, and we would be talking about that. So Monica here is interested in immunoinformatics. Like Monica, are there anybody else who is interested in any other research areas which are not mentioned here? Wonderful. Veronica is interested in neuroscience. We have had a lot of participants who have worked on several projects in neuroscience. So you can actually take a look at some of those projects right here. Let me again post the link. So this link will take you through some of the projects that were developed as part of our community. So Indrani is also interested in immunoinformatics. Shinjini in gene therapy. Sharmin also interested in neuroscience. Okay, thank you for sharing. Okay, Abhay is interested in oncology. So Abhay, we have a lot of resources on oncology. So please go ahead and take a look at learn.omics Logic. So Kanakavali, you're interested in microbial genetics. Go through the metagenomic courses that are there on Learn. I think that will give you a good overview. So Monica here is interested in CRISPR gene editing. Okay, so I will keep the polls open for just a few more seconds and then I'll close it. So thank you all for sharing. If you have any questions again, please write to us. So Shorish here is interested in gene reprogramming. Okay, great. Thank you all for sharing. So I would be ending the poll today. So besides the program that we have mentioned in today's session, as well as in some of our pages, we keep on having many of our programs which are termed under Omics Logic. So please feel free to check out that page or reach out to us with any of your other requests. For researchers and for students who want to go into research and are interested in developing their own project, and necessarily if we do not have a training program on that, we also offer mentor guidance and support through the research fellowship program, which is usually for 3 months or 6 months. So please feel free to go ahead and reach out to us with any of your questions that you may have. And we hope to see you in our upcoming sessions starting next week. So thank you all for joining today. And this video would be available for everybody, both in our YouTube channel, which is the Pine Biotech YouTube channel. And let me share the direct link in the chat box again, for those of you who may want to watch it one more time. And thank you so much, Abhay, for sharing this. And thank you all and have a wonderful day ahead. Good night to everybody who are on the Eastern time zone. So